This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 435, recorded on March 31st, 2017. This episode of TWiV is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get your first 3 meals free with your first purchase and free shipping by going to blueapron.com/TWiV. Hi everybody, I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in New York City, where we have a nor'easter, Dickson, Despommier.
Hello there, Vincent.
Now, you were just outside, right?
I was, yes.
Is it still raining?
It's cold and raining.
Windy.
It's very cold and very rainy.
Pretty nasty.
It's, March goes out like a lion.
Today is the last day of March, isn't it?
It certainly is.
March 31st. And
wait a minute, tomorrow isn't a propitious day because it's the opening of trout fishing season in New York.
4 degrees Celsius here.
Yeah.
I love when the weather app shows it raining. It's a nice little touch. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's 41 degrees Fahrenheit, which means it's 5 degrees Celsius, warmer than you are there. And it is cloudy, and we had all that rain yesterday.
Right.
Yesterday you had it, and it came here, huh? Mm-hmm. Are your flowers blooming?
I saw one daffodil, but not in my yard yet. But the little crocuses and snowdrop things are blooming.
Yeah, we have daffodils. I was looking this morning. Well, I think these are those lilies, daylilies, right? The orange ones, called daylilies. They had all sprouted weeks ago because of that little warm spell. So they're about 6 inches of the palm fronds, those long things above the ground, and then they burn from the cold. So now they're pushing up, but the tips are all burned. But
is that what you called gladiolas then?
No, they're not gladiolas. They're different.
But you called something. You talked about gladiolas. That's what made me wonder back then.
Gladiolas. Yeah, I think I
Well,
messed up. I think I meant daffodils, not gladiolas. Yeah.
Daffodils. Daffodils.
Yeah. As long as they're not cherry blossoms.
And narcissus.
Gosh, if I ever had a cherry blossom in my yard, I would cut it down.
Really?
Oh, didn't George Washington cut down a cherry tree? That was a cherry tree, right?
Put that axe down.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody.
Welcome
How you
back.
doing?
Welcome back,
Thank
Condit.
you.
Oh, that's
Thank
almost
you.
Welcome Back Cotter, isn't it?
Exactly right.
That's almost right. Yeah, it's good to be here.
Yeah, we missed you. Not really, but we'll say it.
Well, I missed you guys.
We missed
We got glorious weather.
You do.
It's
I watched the golf
83 degrees. 83 degrees.
Yeah.
Absolutely, totally cloudless, bright blue skies. That's 28 degrees C. It was like 55 this morning and clear and slowly warmed up to 83. It's been glorious past few days. So there you go. Wildflowers are blooming. Blue bottle. No, not blue bottle.
No, blue bonnet.
Blue bonnet.
Yeah, lupin.
Is a big
They're
deal.
lupin. They're part of,
The blue bottle is a fly, right?
It is.
Yeah.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here, and it is definitely not glorious weather here. It's rainy, foggy, misty, 37 Fahrenheit, 3C, really crummy. We had a little sleet, a little snow.
Right.
Rich Condit, where were you in all these weeks? You're sailing, right?
Well, I started off skiing. Went to Oregon.
Oregon?
With my family from Texas here. They were all on spring break, the kids and the parents. And so we went to the place in Oregon where I have podcast from whence I have podcasts several times. And there's a ski mountain close by. In fact, that's the reason that place was built there. So we went to Oregon and visited some of the local relatives there and went skiing. And then I came back for 24 hours. And on my own, I went to Florida, where there was the St. Augustine Race Week, an annual regatta. And we entered my skipper's boat in that. We had a couple of days of practice sailing, and then we sailed the regatta. And on the tail end of that, I went to Gainesville and visited old friends. And that was just delightful. It was great. One of the best parts about that. Oh, yeah, we went to Satchel's.
Okay.
Yeah. And one of the best parts about that was, I just had a couple of days to hang out there, and I know the people, so I just kind of went from lab to lab and sat down and talked about people's science. It was like being on a seminar visit, right?
Yep.
And I started thinking to myself, maybe that would be a good retirement gig. If you paid your own way, there's no reason that places wouldn't be okay with you just, show up and could you schedule some appointments with me? I want to learn about the science and just go from lab to lab and hear what people are doing.
Yeah, it's called podcasting.
TWiV.
Yeah, it's good.
Could do that.
Science schmoozing.
Now that's what I do. I get invited to do that. Last week, but speaking of invited, last week I was at University of Colorado Medical School, Denver campus, which is actually in Aurora. And shout out to the students who were my hosts, who were all great. And I spent the entire day Friday speaking with students. There's no faculty. We got there and we had breakfast and morning spent with students. I had lunch with students, seminar, and then we went to the local pub at 3 p.m. I said, is it okay for me to have a drink at this hour? And one of them said, yeah, go ahead. It's Friday. And then I went to dinner with students. And so Thank you very much. Next week I'll be at Cornell in Ithaca, New York, doing a similar thing. All right. I would like to tell you about the ASM, American Society for Microbiology, grant writing online course. Learn the best practices in grant writing throughout this course. What you're going to do is get an overview of the funding landscape, more like a canyon, I think. With very steep sides. Learn what
the Valley of Death, I think it's called.
Exactly.
Along the side of the Grand Canyon, there's a 3-foot trail that you can go all the way down on mules. Did you know that?
I didn't know that.
It's so scary. No one has ever fallen, but I will not do it ever. 3 feet? Give me a break.
Right.
You can also just hike down.
You could.
Did you do that?
Yep.
That's a long way.
We hiked down from the North Rim, which is the less visited side.
I don't know.
That's not scary at all. The trail out that way that scared me was in Zion National Park. There's a spot called Angel's Landing, which is up on top of this just really narrow spire of a peak, and going up it, you are right on the side of a cliff, and they've helpfully installed some chains that you can hold on to, but there's no railing or anything. You're just on this little edge, and it's a long way down.
Didn't they build?
This is TWiV on steroids.
What's that?
We start off talking about a grant course, and now we're talking about
hiking.
Isn't there a place in the canyon where they built a glass ledge?
Yes.
20 feet out that you can stand on?
That's true.
The Grand Canyon, yes.
And there's a place in Yosemite called the Mansfields for particular reasons. And you can actually climb up them. They're at about a 35 to 40 degree angle. And it looks okay from a distance. When you get on it and you look back, you're clinging to it like an octopus, right? You wish you had 8 more arms to just hang. It's really unnerving to realize that if you just made a false step, you'd tumble all the way back down. But again, I don't think anyone's ever been hurt on that one either.
Which is very much like the grant writing process. So
yes, exactly.
What can people do to climb up that cliff?
Well, if you
ask Sisyphus.
If you take this ASM grant writing online course,
you'll
learn what makes grants successful and get personalized feedback on your grant. It's a 7-part series taking place in the fall of 2017. You have to register. The deadline's July 15th. You can learn more at bit.ly/ASMGWOC17. Those are the digits, 17. ASMGWOC17.
That would be ASM Grant Writing Online Course.
Exactly. Very intuitive.
Yes.
We have a few follow-ups here. First one is from Sam. Hi, TWiV team. This is not technically follow-up, but it's close, and it's worth it.
It's informative.
I just listened to TWiV 433, and while I was very interested to learn about antiviral mechanism, so I've learned that when people say while, the clause is followed by but.
Yes, that's right. There's a criticism coming.
While I was very interested to learn about antiviral mechanisms in my favorite nematode worm, I have to make a few points. It's kindly said.
Yeah.
C. elegans is a species of self-fertilizing hermaphrodites with a very low incidence of males. While there are some male-female Caenorhabditis species, such as C. afra, this is not the case with C. elegans. N2 is standard wild-type strain of C. elegans, first isolated by Sidney Brenner. Nematode growth medium, NGM, is essentially a non-selective solid bacterial medium which is generally seeded with the E. coli strain OP50, which the worms eat. The worms cannot eat the NGM itself, and once the bacterial lawn is eaten, will starve. They will adopt the dower stage and can survive without food in this way for many months.
Many months.
A big fan of the show. Keep up the excellent work, Sam in Toronto, Ontario. Thank you, Sam, for the points, which we would not know, right, Dickson?
Mm-hmm.
Well, I should have known. I'm a nematologist by training, but I work only on sexually dimorphic nematodes.
A person on the street would say, typical scientist. Look, he works on sexually dimorphic.
Well, we're dimorphic organisms, I hope.
Yes.
Yeah.
For the most part.
So, C. elegans is infrequently sexually dimorphic.
That's
right?
Exactly.
And I don't work on either of those.
No.
Alan, can you take the next one, please?
Sure. Steve writes, "Hi, Vincent et al. Interesting program this week. I love all the carefully thought up entomological and morphologically descriptive words. They're so much better than the alphabet soup being used to describe biochemical entities." Yes. Good descriptive names for the various parts and life stages enable different species to be compared so that you can see how evolution acts on each segment of, say, the fundamental segmented worm to gradually develop the increasing complexity of all the vast variety of arthropods. A good name for each structural component of the basic bug construction kit makes it possible to describe very complex creatures with great accuracy on a bug blueprint so that readers know exactly where to look to see what you want to draw attention to. Mm-hmm. I'm not an entomologist and I've not read the paper, but when I hear you wondering if secondary sexual features is a mistake, I can imagine that if you are dealing with something that affects the development of an insect into functional or sterile males and females, you might want to distinguish between the functional females and the phenotypic females that can't reproduce, as in the case of honeybees, for example. Your authors probably mean to make sure that they can determine the effective sex ratio separately from the outward sex ratio suggested by the visible phenotype following each phase of the experimentation. That would be my guess if it isn't a mistranslation. If you want to see some more brilliant words, you might like to check out the wiki on screwworm flies, which you could then cover in TWiP, as the outbreak in Florida has just been brought under control by a successive release of sterile males, which works in this species because the females only have one breeding cycle.
Right.
I'm sure Dickson would relish describing the gory details of the infestation by the live flesh-eating primary screwworm as compared with the dead flesh-specializing secondary screwworm, which is also useful in estimating time of death in found corpses and even cause of death, where poisons are found in the insect's guts. Then Steve continues, knowing Kathy's love of Lego and now hearing that she says she's never seen Alien, you might like to get her interested in the idea by showing her this famous scene from the movie. And she clicks to a YouTube video that is a Lego recreation of the scene in the mess hall where the person who was previously had a face hugger on them has it hatch out. So that's classic.
They even have a video where they compare it to the Lego in the original.
Yes.
The chest burster.
Steve is in Bedfordshire, England.
I will try to check it out.
Sorry, where spring is sprung, the clocks have just gone forward, and wild plum blossom is ablaze in my overgrown garden.
Nice.
Oh.
Yeah, you should watch the
yes.
YouTube, Kathy, because that's really the essence of it.
Okay,
that's good. Dickson is not the only one with an interest in screwworms, so I've provided a couple of links here to the Walter and Ina letters archive. My grandfather worked extensively on that. In fact, he was working on it when he met Ina, who he married. And I just noticed when I looked this up on that site, one of his letters from 1940, I think, or 39 or 40, he's commenting, I also have the feeling we'll get somewhere on the screwworm problem through Cushing's work. And Emery Cushing was one of his colleagues, actually one of the people he supervised. And Cushing is best remembered for having developed the sterile insect technique for screwworm.
Right.
which is exactly what we were just discussing. So that is the origin of that work, and you can go read letters from the person who was supervising it.
Dickson, why don't we do a screwworm TWiP and have Alan come on and talk about it?
We'd love that.
Sure.
What do you think?
You bet.
That'd be fun.
All right, we'll plan that.
Sure.
TWiP is a fun one too, right, Dickson?
Oh, yeah.
So this week on Twitter, Stephanie, who was on TWiV some time ago, her Twitter handle is Mucus Matters.
Nice.
She wrote, the end of this week's TWiV was great. I want end of TWiV sassy Kathy throughout the podcast. She is not sassy enough for my taste.
Yes.
Throughout the podcast.
Yes, I will try.
Ramp
But
it up.
also another Twitter person called me exasperated.
That's not me.
Yes, no.
Fake Despommier
Yeah, not me.
is speaking in your
I never said that.
ethos.
Right. This was about all the stuff that we were talking about the secondary sexual characteristics and the secondary thing that had nothing to do with that was in the paper. Yeah.
It was this,
And I had to go back. Yeah. I normally don't listen to episodes that I'm on because I already was on the episode. I only listen to the ones that I'm not on. But I saw this comment and I said, oh, this must have been, I don't remember this. And it turns out it was when you all were talking, waiting for me to show up.
Mm-hmm.
Right.
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 I didn't find it all that sassy.
Well, you've heard me before. You've heard me.
Well, I think, my wife has kind of calibrated me on that. So, it takes a lot to move the needle on sassy for me.
Okay, well, we'll see what I can do.
Rich, are you there, Rich?
I'm here.
Get close to your mic.
Oh, I'm right on top of my mic. Did I go away?
Yeah, you kind of
A little
faded
bit.
away.
You
Can
faded a
you
little
take the next email, please?
Okay. Jim Ho Lee writes, Dear Vincent et al., I thoroughly enjoyed your discussion on parasitoids. Everyone was friendly and made the discussion fun. At the time of this writing, it is a dreary 40 Fahrenheit. There is still snow everywhere. I'd like a chance to win that book. You guys are the first science podcast I have ever listened to. As a millennial, I normally consume my science via YouTube videos, most notably SciShow. I'm a senior molecular biology at Skidmore College, taking Dr. Hilleran's virology, RNA, and biology of cancer classes. So there's a fair amount of overlap between the classes. In China, they've already begun genetically engineering 3N human embryos to cure lung defects. It gives a link. What are your thoughts on genetically engineering viruses to skew the sex ratio in the other direction as a way of improving pest control? There is a C2 CRISPR-derived system that can edit targeted mRNAs. And gives a link to that. Sincerely, Jim Ho Lee Lung. P.S. In traditional Chinese symbolism, yin-yang, each swatch contains the seed of its opposite. I thought it was a clever play on symbolism where you would have the seed of male skewing viruses inside of the females. Not sure how intended that was. In standard Mandarin pronunciation, song is pronounced sung, and Ji is jug, like your soft grunting. He's writing out pronunciations for these. He is hug. I really appreciate that you guys try to get the pronunciations correct.
Even for the word.
So was right, exactly.
Yeah. So was the I didn't look at this. Was the yin yang symbol in the authorship?
Yeah, first two authors.
Done weird.
No, it was normal.
No, I don't think so. It was just that it was a symbol for the co-authors.
And we found it on this week's TWiP paper, too, remember?
We did.
And so it's a standard thing at PLOS, I guess. PLOS pathogens, anyway. Now, what about using viruses to skew the sex ratio in the other direction? Does that make any sense?
If the CRISPR allows it to happen, why not?
Yeah, you have to figure out how to do it, right?
Exactly.
Right.
And the way the viruses do it, we don't know, but we could figure that out. Maybe we could then do it the other way. Not so straightforward, I think. Go for it. Go for it,
Exactly.
Jim Ho Lee.
You may just change the world.
Skidmore is up north, right?
Yep.
I knew,
Saratoga Springs.
Saratoga Springs, right.
Right next to the racetrack.
The New York City Ballet goes there in the summer.
It's true.
Oh.
Okay.
Yeah, they have a music camp for kids up there, too.
I like Saratoga Springs. It's nice.
Yep.
Maybe I could do a podcast right there in town. Get a little studio,
Yeah.
Every little town I talk about.
Did you ever visit the springs, Vincent?
No, I never visited.
That's too bad because they're sulfur laden.
Sulfur laden?
Oh, yeah,
Do they have sulfur in them?
they do. They're supposed to be health giving, but a little bit of a pungent odor.
Okay. All right, we have a snippet for you and a paper. And the snippet is from an old friend of ours who's been on TWiV. And this is a PLOS Pathogens paper called The Blood DNA Virome in 8,000 Humans. Actually, 8,240.
Right.
So why wouldn't you put that in the title instead of 8,000? It's over 8,000, right?
It is over 8,000.
But it's not over 9,000.
It's not over. Wow, Dickson, I'm so impressed.
You just, you put me onto that, and now I can't get it out of my head.
It's over. What was the eosinophil count?
Yeah.
All right. The first author, who is a sole first author, is Ahmed Mustafa. And then we have G, Kirkness, Biggs, Wong, Turpaz, Bloom, Eric Delwart, Nelson, Craig Venter, wow, and Amalio Talenti. Now, I was just wondering, why does Craig Venter have to be on papers anymore?
Because he paid for them.
Yeah, but
He funded them.
at some point, you've done so much, just let other people be on the papers, right?
Well,
And even, remember
Craig
John
Venter
Stites?
seems like somebody who probably greatly appreciates having his name on things,
Yeah, he does.
given
Yeah, it
that
says
he
he-
runs the Craig Venter Institute.
He conceptualized it. He was important for the funding acquisition. He was involved in the supervision,
this is true.
according to the author contributions listed at the end of the paper.
I just want to bring up Joan Stites, whose mentor, Jim Watson, let her publish on her own. Okay?
Yep, and she did the same for her students.
And I would argue that Jim Watson was quite prominent in
Very
his
much.
day. He's still alive, of course.
Even more prominent
You think?
than Craig Venter. Yeah, sure.
It's hard to compare, but Watson did have a Nobel Prize, right?
He did.
And he did discover the structure of DNA, or sorted it out, co-discovered.
It's true.
Dickson, did you co-discover Trichinella?
No.
Okay. All right, this is
So there are 11 authors on this paper.
Yes.
Paget and Owen
Of those,
co-discovered
Eric is from what's it called? The Blood Systems
System.
Research Institute,
Correct.
and Craig is from he's got
He's got his own
affiliations
institute.
Well, actually, he's got his own institute, but the primary affiliation of the remaining authors and all of these other guys is Human Longevity Inc.,
Yep.
which is a San Diego-based venture launched by Craig Venter and Peter Diamandis in 2013. Its goal is to build the world's most comprehensive database on human genotypes and phenotypes, and then subject it to machine learning so that it can help develop new ways to fight diseases associated with aging. So they're into sequencing and using the sequence, specifically human genomes.
Good for them, right? Anything
Good for them.
works. I'm happy about it. Well, what they did here is they had sequence data, whole genome sequence data from 8,240 individuals. None of which had any known infectious disease. At least they didn't have any symptoms of infection.
That's a better way to put that.
Yeah, and
I was a little perplexed by the way they put this in the abstract. None of whom were ascertained for any infectious disease.
Oh, that means we didn't check.
So that means they didn't have it, or you didn't look, or you didn't see anything obvious. I'm not sure.
Exactly.
Well, anyway, these genome sequences were done, and then they decided afterwards to see, could we take this information and find some viral sequences in it?
And importantly, the source of the material for doing the sequences is blood.
Yeah.
From blood banks.
And it's blood cells specifically, right? Which you can't find in the paper, but it really bugged me that they don't say where they got the DNA from. That's you're supposed to be able to reproduce a paper. Not that anyone would reproduce.
Not that anybody can reproduce.
That'd be tough.
But you should know. And so Eric Delwart says it's from blood cells, which is important, as you will see. So they had these sequences. They had 1 petabyte of sequence data. Now, how many zeros is that?
Many.
A lot.
Google's worth now, not quite that many, but a lot.
It's a lot.
It's a lot.
Actually, I did write about it.
100,000 runs from smaller mini Illumina machine sequences.
Oh, Dickson, you're getting technical.
I'm reading this.
3, 6, 9, 12, 15 zeros in a petabyte.
Thank you very much.
Wow.
They took this 1 petabyte of sequence and they did 500 billion similarity searches on their iPhone.
Of course. No, not really.
I'm waiting for people to laugh. No one laughs at me anymore.
I got it.
That's all right.
Well,
You
I
don't
couldn't
have to-
figure out if it was April Fool's Day or
This tomorrow.
joke. I know, yeah.
Anyway, so they're looking for viral genomes, and they found some. They got sequences of 94 different viruses, including 19 human DNA viruses and some proviruses and a few RNA viruses, which was interesting. They found herpes viruses, anelloviruses, small single-stranded circular DNA viruses, papillomaviruses, polyomaviruses, adenoviruses, HIV, HTLV, hepatitis B, Hep C, parvovirus, and influenza virus. And these were in 42% of the participants. All right. And just a few interesting tidbits. Merkel cell polyomavirus in 49 people, papillomavirus in 13, parvo in 6, and herpes virus 8 in 3.
And of course, when you see results like this, one of the first things you ask is, how much of this is contamination?
Right. Well, they had 75 other viruses, which they think were contaminants, including some giant viruses.
I would
And phage.
think so.
Phage.
That's right.
They have a lot of, most of them were contaminants, in fact, from probably the preparation of the blood, the preparation of the samples, the sequencing, right? Because this is very sensitive, and so it's easy to be contaminated. But so 75 contaminants, 19 human viruses. Now, 42% is a low number, and that's because these are blood cells. They threw away the liquid phase, right?
Right.
And in other studies, people have found anelloviruses in everybody, in every blood sample you look at, as long as you look for the in the liquid, not in the cell. So when you look at the cell, you're looking at viruses that are either in the cell or on it, right? So you cut back on what you're looking at. And, of course, there are no RNA viruses here because they're doing DNA genome sequencing, so there's no reverse transcription step. The Hep C that was found happened to be integrated. A piece of it was integrated into cellular DNA, which happens, with RNA viruses. Mm-hmm. And these retroviruses were present as proviruses, HIV and HTLV. So they had integrated into the cellular DNA. All right, so that's really what they found. It's, I guess, maybe the most important thing here is that there's a lot of contamination. Or as Eric once said to us, DNA is a problem. It's everywhere.
They're everywhere. And they talk a good bit about how a lot of that comes from the reagents.
Yeah.
Yeah, that was going to be my question, is what are the major sources of it here?
I think a lot. Yeah, I think reagents. Let's look at some of these contaminating viruses that they found. They have lovely graphics here. Viral integration. Yes. Some of the giant viruses I thought were funny. Yeah, Mali virus, chlorella virus, Mumu virus. These are viruses of amoeba, we think. So they're probably contaminants of water at some point, maybe. And they're pieces. They're not entire genomes, remember. Did they find any entire genomes? I don't recall.
They got, I don't recall, but they've got this figure down here where they talk about reconstructing them. But it wasn't clear to me whether those were reconstructions out of individual samples. Probably.
Yeah.
And looking at this, they've all got gaps, except maybe, that they had trouble getting the ends of the parvovirus. But they've all got gaps in them. Yeah. I don't know that they got a total sequence.
Is the title of this article a little misleading then, to say virome?
It's not the complete virome, yeah.
No.
Well, yeah, I think it's misleading. I think that they should just say, viral sequences found in human blood samples.
Any ohm that you're looking at is going to be incomplete.
Yeah, I guess so.
Yeah, but if they do the human microbiome of the gut tract, for instance, they're dealing with whole organisms to start with.
Yes.
Here, they're not.
Yeah, you probably didn't get all of them.
Well, as you'll see from Eric's letter, this was a study of convenience.
Right.
Yes.
Which,
sure.
Ohm in this case does not imply that you got the whole sequence of anything. It implies that you're looking at how many different viruses, all the different viruses, the collection of viruses.
I think the omics suffix is usually used to refer to the breadth of what you're looking at rather than specifying a particular level of depth. Like they're trying to look at as many, to find as many different viruses as they can in as large a sample of people as they can.
And they use stuff they already had. They have data that they already had. That's the whole point here. They didn't go out and do this from scratch. That's kind of a pun, isn't it? From scratch.
A little bit.
From scratch.
All right, so this is interesting. Currently, we test donated blood for HIV 1 and 2, HTLV 1 and 2, hepatitis C, hepatitis B, West Nile, and Zika virus. That's it. And so these other sequences they found, they don't know if we should worry about them. And, an RNA virus that's highly prevalent in blood is a PEGI virus, GBV-C also called. These anelloviruses are in everyone, and we don't think they're pathogenic because they're in everyone. So we don't reject any blood based on their presence, otherwise we would have no blood supply. Right. But, the parvoviruses they found here, they say we don't know if this is a concern, and other DNA viruses. So I would say, a virus has to be shown to be transmitted in the blood, otherwise shouldn't worry about it, right?
Exactly.
You find pieces of genomes.
Well, that's why I was questioning the title a little bit.
Yeah, I understand. Right.
But this also kind of establishes a scope of what to look at when somebody claims to have discovered a new virus that's implicated in some condition. Mm-hmm. And they point to the XMRV example and a couple of others where we've had viruses, this might be the cause of this, and it turns out, well, no, that's just kind of there. And here you have finally the largest sample to date of looking at a whole bunch of DNA viruses and finding that a whole bunch of them are already there. So you could easily imagine if you didn't have this baseline, somebody might go and say, human herpes virus 4 causes thus and such chronic disease because they found it in some large percentage of the population, but then come to find out that it's just normally in a percentage of the population.
Yeah, good point. So here we are at TWiVome, actually. Would you agree, Dickson? We're at TWiVome?
But you have the complete sequence on us, though.
Because Eric Delwart said, Dear Vincent, and the TWiVome.
The TWiVome.
This study shows how human genomic data can be repurposed or mined to analyze a part of the human virome. There you go.
Part of.
To put the scale of the analysis in context, the 1 petabyte of data used here is the equivalent of 100,000 runs of the smaller MySeq Illumina machine. The amount of data restricted the analysis to finding viruses using nucleotide sequence similarity only rather than the computationally much more demanding translated protein similarity searches needed to detect divergent, i.e. new viruses. The focus was on a small fraction of that starting data, 0.000005 that matched viral sequences. As most of the people were likely healthy, the viruses detected, with few exceptions, typical commensal blood-borne viruses. The focus on blood cells left out the large majority of human viruses that are usually restricted to respiratory, skin, or enteric tissues. Because of the way human genomes are sequenced, the viruses detected were largely limited to those that are associated with blood cells and have double-stranded DNA genomes, or at least form a double-stranded DNA during their replication cycle. The dominance of the herpes viridae reflects the focus on cellular DNA rather than plasma, where these viruses are more rarely detected. Anellovirus were also commonly detected despite their single-stranded circular DNA genome, likely because they were caught in the act of replicating as rolling circle double-stranded DNA intermediates.
Caught in the act. I like that.
Caught in the act, yes.
As expected, the very common blood-borne RNA PEGI viruses in the flaviviridae were not detected. As you often state in TWiV, finding viral genomes does not prove that the virus is infectious. Thank goodness someone is listening. Sequences from viruses known to replicate in bacteria, algae, amoeba, or fungi were also detected, likely reflecting DNA contamination in the many reagents used to generate human genome libraries. Human tropism for these viruses has not been shown. Papillomaviruses and polyomavirus sequences were also detected despite their known tropism being restricted to epithelial cells. Their detection in blood may reflect either other sites of replication or sample contamination with skin cells during phlebotomy.
Yeah, I like that part.
Yeah, but we also slough lots of endothelial cells that end up in blood, so that's not unusual to expect that to be there.
So, despite the bias of the starting material, blood cells focus on double-stranded DNA and lower sensitivity relative to targeted PCR studies. The study shows that analysis of raw human genomic data can reproduce the known epidemiology for some viruses, like the higher prevalence of HTLV proviruses in people of African ancestry, which we didn't talk about. If similarly analyzed, the growing amount of raw human genomic data will allow large-scale epidemiology for blood cell-associated double-stranded DNA viruses. Data from the major human populations located in either their ancestral or new homes may facilitate studies of the relative roles of these infections of human genetics versus geography and lifestyle. By combining the human genomic and viral information, the identification of human genome polymorphisms associated with particular infections or high viral loads will also become possible. Yes, so that's the power here. You can use this going forward to look at all these things. Lots of these analysis would depend on available computing power. Maybe Facebook, Google, or Amazon will have some to spare.
I was kind of surprised to see that because I had assumed that they would have used what have now become kind of standard tools, the Amazon S3, which is their storage service, and EC, Elastic Compute, which is their rent an Amazon CPU cluster service. A lot of big bioinformatics projects already use those.
Well, you have to pay for them. Maybe they don't want to pay.
You do, but they're priced.
They're pretty.
They're priced
right?
very competitively, and the important thing is that you don't have to buy them. You are renting them.
Yeah, exactly.
So you use what you need while you're doing the project, and you pay only for what you use. After you're finished running your programs and doing your analysis, you don't owe them anything. Whereas if you build some computing center, you've got to maintain that and hire people to update it, and then it gets outdated, and it's just. But I don't know. Maybe since the Venture Institute does so much of this, they probably do a lot of their own stuff in house.
I'm sure. Yeah, and they probably raise their noses at Amazon EC.
At Amazon, yes. And at a certain scale, it would make sense to own your own if it's really getting full use out of it. Yeah.
Well, I'm sure Venter and Bezos talk all the time, right?
Probably.
The billionaires do that.
Or they refuse to talk to each other. I don't know, maybe.
No, I think they do talk. I think you're right.
Okay.
Vincent.
Yes, sir.
In Figure 4, you have the chance to talk about one of your favorite viruses, and you didn't mention it yet. The very last one.
Hang on, let me get it back. It went away.
Yes.
Figure 4. Hang on. Figure 4. That's Figure 6. Sorry. 4. Torquetano. Is that it?
Yeah,
yes.
That's it.
Yeah, Torquetano, TTV. I love it. One of those little single-stranded DNA viruses that are in so many people.
Must have also been caught in the act.
Torquetano. Yeah, it was caught in the act. That's a good title. Caught in the act.
Torquetano in flagrante.
Thank you very much for mentioning that. Cannot resist.
I'd rather have tortellini than tortacano.
Well, speaking of eating, Dickson.
Yes, I thought I would bring that up.
The sponsor.
No pun intended on that one either.
Sponsor of this episode is Blue Apron. Their mission is to make incredible home cooking accessible to everyone in the continental U.S. while supporting a more sustainable food system, setting the highest standards for ingredients, and building a community of home chefs. As far as I understand, they don't go to Hawaii.
They probably would like to do a larger scale than that. But
or Alaska.
Yeah, but
this is what they're doing now.
Alaska is, but that's not continental, but it is attached to the continent, right?
It's food desert. It's a big food desert.
Yeah. What they do is deliver seasonal recipes along with everything you need except salt, pepper, and oil to make delicious home-cooked meals. I know they don't give you salt, pepper, and oil because I had to find my own, which we have. It's not a problem. But a lot of the other things you might not have, you don't want to go buy a big package of saffron, for example.
Right.
If you only need a little bit. And they give you, if you need soy sauce, 2 tablespoons, they give you these tiny bottles, pre-proportioned. Everything's wrapped up nicely. It's very, very nice. If you like everything all wrapped up tidily with a bow, And they also give you a recipe card, which is beautiful, nice photos you can keep and redo at your leisure because you won't get that same recipe in the same year. So if you want to do it again, you could go buy the ingredients and do it. And 40 minutes or less to make the meal and less than $10 a person, which is a really good price because they don't have stores. They have contracts with over 150 farms, fisheries, and ranchers all over the U.S. whom they buy from, and then they have warehouses where they package everything together. I just learned that they're opening a new warehouse in Linden, New Jersey, which is a mere 20 minutes from my home, and next to Rahway, which is where Merck is,
and next to Newark Airport.
Next to Newark Airport, and right there in Linden. Linden is famous not only for its pierogies, for the Exxon refinery, which gives all of New Jersey a bad name.
And a bad smell,
too. I find that the Bayway refinery kind of pretty at night.
Oh, at night it's amazing. All those cracking
when
towers,
you're driving
right?
past on 95, and it's all lit up, and it's just this massive industry. And yes, it's had problems over the years, but it's just kind of cool. And it's there next to Newark Airport, and all the railroad cars and the big tanks. It's neat.
The burn off every now and then from the towers. So if you land at Newark, you often will go over it too. Right. It's quite an amazing structure.
Mm-hmm. There's also an airport right in Linden.
There is.
There is. General Aviation Field that's right in among the oil tanks.
Have you landed in that, Ellen?
I have not yet, but I've flown past it, and I may go in there at some point if it's convenient to where I'm headed.
There used to be a big GM production plant there, which they closed and made a big mall instead, right next to the airport as well.
Right.
So anyway, they're going to be. What they do is they collect from seafood. Where do you get seafood from, Dickson?
From
farms,
the sea.
Right? No, not from the sea. From farms. No, you don't
Depends. Sometimes wild caught salmon is available.
Yeah,
but mostly it's farmed fish.
Tuna as well, right?
Tuna is wild caught.
And trouts can be farmed, right?
Trout are farms.
Yeah,
for sure.
And also tilapia.
Tilapia is farmed.
Yeah, we talked about a tilapia virus here on TWiV,
and
but they don't have any of that on Blue Apron tilapia. Don't worry about it. Beef, chicken, and pork from responsibly raised animals, produce from farms that do regenerative farming, and they give you just what you need, so there's no waste and you don't overeat. Unless, as you do like Dickson, he orders a meal for 4 and eats it all himself, right?
Exactly.
Don't you love it?
I do my own cooking, thank you.
You do?
I do.
But you do Blue cooking Apron. With this. This is cooking.
No, we've never used Blue Apron.
So you feel the prep is all part of the cooking?
No, the shopping is fun.
I hate that part.
I love it.
I hate it.
Sorry.
I hate it.
Well, it's personal.
No, I don't hate. Hate is too strong.
Hate is too strong. I just like,
because then you get home and you forgot something.
No, we enjoy selecting our produce.
Good. Well, you do that. Me, actually, I love Blue Apron. I think it's great.
I will try it sometime.
And you can customize your delivery, your dietary preferences, whatever you want. And every year, they don't repeat. Whatever they have at any particular time, it's not going to be repeated again throughout the year, so you're always surprised. This month, brand new recipes here. Spinach and fresh mozzarella pizza with olives, bell peppers, and ricotta salata. Ricotta salata. That's the hard ricotta that you can grate.
Speaks.
Not the liquid one, not the soft one. Sweet and sour salmon with bok choy, carrot, and ginger fried rice. Baby broccoli and fontina paninis with hard-boiled egg and arugula salad. My mouth waters.
Well, look at him. He's salivating all over his shirt. You
no,
can't
I am
see
not.
this, but I can see this.
Stop it.
He needs a bib every time he reads this.
Be nice to Vincent. Be nice to Vincent.
I am being nice to you.
I'm not salivating on my shirt. If I were, I would.
No, you're living the ad. You're living the ad. Come on.
Well, that's what they want.
Of course.
Blue Apron, that is.
You bet.
Check out this week's menu. Get 3 meals free with your first purchase with free shipping. Go to blueapron.com/TWiV. If you go there, you get a page that says This Week in Virology at the top. Whoever knew that Blue Apron would have a virus on its front page?
Yeah.
blueapron.com/TWiV. You'll love how good it feels and tastes to create incredible home-cooked meals with Blue Apron, so don't wait. Blue Apron, a better way to cook. Mm-hmm. Isn't it cool that our guest a couple of weeks ago, Brianne Barker, she got turned on to Blue Apron by TWiV, and she uses it all the time.
It's nice, of course it is.
It's very nice.
You bet.
We thank them for their support. Now we have a paper for you, published in MBio. There are no yin-yang symbols on this.
No. And by the way, both the papers today are open access. Mm-hmm. You can go and read them at your leisure.
So go now and get it. You can read along with us. Oh, if you're driving, don't do that.
That's right.
No, don't do that.
MBio, plaques formed by mutagenized viral populations have elevated co-infection frequencies. Mm-hmm. This paper was suggested by Catherine R. Spindler. Is that right?
That's correct.
R. What does the R stand for?
Ruth.
Ruth.
Mm-hmm.
Nice.
Ruth Chris, right? That's a restaurant chain.
Yeah.
All right, Elizabeth Aguilera, Andrea Erickson, Palmi Jesudson, Christopher Robinson, and Julie Pfeiffer, who are in the Department of Microbiology, University of Texas, Southwestern Medical Center in Dallas, Texas, hundreds of miles from Rich Condit. Where are you, Rich Condit?
I'm in Austin, Texas.
You've been quiet for so long.
Well, you guys are just yakking like crazy. I can't get a word in edgewise.
Oh, you of all people.
Yeah.
Welcome to my world.
No, you're just retired.
There's the kind of thing we want.
I don't have anything of value to say.
About food?
We don't believe you.
I wouldn't want to break into a commercial. I mean, that's sacred territory.
No, I think they like when people break in.
They do. They do.
Yeah?
They do. Yeah, sure.
Okay.
So this is all about a plaque assay.
Mm-hmm.
Now, before we talk about the paper, why don't we let Kathy describe a plaque assay? She likes
Sure.
it.
Only
The
if
very
she's.
first
Oh, wait a
plaque
minute.
assays-
Only if it's sassy.
No, the first plaque assays I did were Phi-X 174 plaque assays. They're done a little bit differently, but I'll tell you about animal virus plaque assays that are done here. For that, you plate cells in a monolayer on your plate, and you want them to be a certain amount of confluence. That's a certain density of cells per plate, depending on the virus that you need to assay and maybe how long the plaque assay has to last. Then when you're ready to do the plaque assay, you make dilutions of your virus stock, usually 10-fold dilutions, and you remove the media from the plate and you put your virus stock on, your various dilutions on different plates in a small volume, and you let that absorb for usually an hour. And then, depending on the experiment, or the lab custom or whatever, you might remove the inoculum or you might leave it on. But in any case, you overlay it with a mixture of media and agarose so that the viruses that burst out of a cell can't swim around in liquid media. That's semi-solid media with the agarose. And so traditionally what we say is that wherever one plaque forms is where one virus, one infectious unit has landed. And so you can imagine that if you have your virus stock concentration too high, the dilution is not enough for what you put on the plate, you'll just totally wipe out the plate. And if you have it too low, you won't see any plaques. And so you want the Goldilocks amount, which depending on your plate size is usually somewhere between 20 and 100 plaques per plate. So that's a nice countable number. Lower than that, it's lower than 20, it's probably not reliable. Higher than 100, you might start having a lot of overlapping plaques or not be able to see all your plaques very well.
So, Kathy, I have a few questions because I've never done a plaque assay. So
Okay.
This is from the outside world. When you dilute your virus, are you diluting it in medium?
That also depends. In my lab, we dilute in PBS, mostly because you don't need to have the medium and it's cheaper to use PBS than medium.
Okay. And the amount of fluid that you use, is that equivalent to a certain millimeter level of completely covering the cells so that when you swirl the plate, you get an even distribution of the fluid? Is that how you do it?
Right. So when you do the adsorption, you want to have enough of a volume so that you get it overlaying the whole plate. In fact, we do a rock forward and back and then a rock left and right to try and distribute it. We don't want to set up a circular motion and spin everything out to the edges.
That solved a big mental image for me.
Behind you, there's a whole stack.
I helped you put that up, actually.
You can see, just look at it.
Right.
Now, on TWiV 68, if you search Google for Ode to a Plaque,
Yes.
first hit is our TWiV number 68. Vincent, Alan, and Rich are enthralled by movies of vaccinia virus plaque formation.
Those are fabulous.
There was a movie made by
great.
Jeff Smith and his laboratory over in the UK, so that's a good thing to look at. I also did a virus watch on how to do a plaque assay. It's a movie.
And I think we also talked on TWiV 145, the Invincible TWiV. Rich and I talked
Had the
about
first one, yeah.
Yes, Twort and Darrell and their work, and I think they did the first. One of them did the first plaque assay, didn't they?
Darrell.
That's right.
Darrell did the very first plaque assay.
Dickson, you like that name? The Invincible TWiV?
Love it.
You get it?
Love it. I do.
I wasn't there.
No.
That was the only one, I think.
We've been,
No, we did another. We did one with when Dickson was on. We did Jenner's
That's right.
Inquiry.
Right.
Twice.
Yes.
What'd you
Me
call
and
that one?
Alan and Dickson.
What'd you call that one?
None of Vincent.
Recall.
Well, at least for animal viruses, way back in 1950, what year was it?
53.
The year I was born. So that year, the structure of DNA was discovered. Plaque assay for animal viruses was published, and I was born. So this is all why I'm a virologist.
This has to.
And whoever else
mean
was born in
something.
50. Kathy, were you born in 53?
I was.
So Kathy and I are virologists because of these coincidence of events.
I love it.
I tell my classes this all the time, and they roar. With the only thing they ever roar with laughter. They think it's so funny that this old fart is talking about why he's a scientist. That year, Renato Dubecco and Marguerite Vogt published a paper, a very short, simple paper, Plaque Formation and Isolation of Pure Lines with Poliomyelitis Viruses. It was before we called it poliovirus. And they developed a plaque assay based on phage plaque assays for animal cells and poliovirus. And they showed the dose-response curve, if you make dilutions of virus and plaque them out, is linear, which they concluded means that one virus is sufficient to initiate a plaque.
One plaque, one virus.
So that's what they re-examined.
One gene, one protein.
They re-examined in this paper using different methods.
Yes.
Is there more than one virus in a plaque?
So,
By the way, TWiV 170 was the other Vincentless TWiV from Viralis effluvia to VLPs.
So, a question to Kathy again. Then, if I'm just learning this for the first time, and I've got my plaque assays all set up, and I've got my overlay, and I'm golden now, and I put them in the incubator at 37 degrees. When do I look?
Depends on the virus.
So tell me which virus does which to win.
Okay, so I'm not sure how long poliovirus plaques take. I know that
Vincent
VSV
knows
plaques,
exactly how long.
VSV, you go overnight. For human adenovirus, we go 8 days. We start seeing plaques, and then we continue to count over the next several days.
Wow.
I think some polyomaviruses take almost 3 weeks. Is that right, Rich?
Mouse polyomavirus took 2 weeks.
That's incredible.
Yeah. Okay.
And as a matter of fact, we used to do a second overlay to refeed the monolayers.
We refeed the adeno ones every 2 to 3 days. And phages, I was going to tell you, since that was my first plaque assay, those develop in 2 to 3 hours.
Exactly.
When we did T7, we either put them put them at 37 degrees for a couple of hours, or if you were doing your thing at the end of the day, you could just leave the dishes out at room temperature on the bench and get plaques the next morning. And if you put them in the incubator overnight, you wouldn't be able to read them the next day because the plaques were too big. Vaccinia takes about 3 days, 2 or 3 days, but I used to routinely let them go a week because I didn't want to miss anything, in particular since I had viruses that form different sizes of plaques and that kind of. I like big, fat plaques.
And also, Dickson, depending on what you want to do, you can stain your plaques. John Holland published a paper showing staining with crystal violet. You can also stain with the agarose still there with crystal violet, or you can take the agarose off. But you can also stain with the agarose still there with neutral red, and that makes your plaques kind of turn yellow, and the rest of the media stays kind of an orangish color. And that kind of is only good to count the next day. A couple days after that, the colors all kind of blur and you lose that.
Are plaques easy to harvest for organisms out of the same plaque? I mean, are they easy to get into and come back out again?
Yeah. Take a pipette and poke it right into the plaque.
That's the basis for whether there's
so
one virus or two viruses
plaque
in the same
purification,
plaque. Yeah, got it.
If you want to purify virus out of a plaque, then you got to do an overlay with something like neutral red, because the crystal violet staining conditions usually kill everything.
Exactly.
Of course, with polio was 2 days.
2 days.
And we can see the plaques without any staining, so we can just pick them. We circle them and pick them.
Nice.
Right.
You get
Same with adeno.
You get a little plug of agar. And the overlay depends on the virus. So for some of our viruses, we use back-to-agar, which is pretty crude. For others, we use agarose. For others, we use different purified forms. It's very interesting that the viruses are inhibited by different things in agar, depending on how it's prepared.
As the plaque grows,
Yeah.
can you make a correlation between the size of the plaque and the number of viral particles per plaque?
As it gets bigger, there's more virus because multiple
has that been
rounds
done so you can get a quantitation?
You could. I haven't done it, but you could pick bigger and bigger ones, and there should be more viruses because you've gone through more rounds of replication, right?
Sure.
Right. More often, the plaque assay itself is your quantitation.
Yeah, sure.
Okay, so you do it either to plaque purify, and we do it 3 times to make sure, as you will see in this paper, which kind of emphasizes that, and to quantify the titer, right?
So on a micro scale, another question. If you're observing this under a microscope in a 37-degree room so that you can maintain the culture at the same time, when is the first cell from this inoculation? When does that first cell die?
That depends on the virus. In fact, in that movie that was made,
they to a plaque.
They looked they infected, and then they looked to see the first cells that were becoming refractory, which indicates their infection. Then they put the camera on them and did time lapse.
Got it.
And you can watch the wave move out.
Do you remember that movie?
I don't actually.
It's
I'm going to go back and review it.
It's on YouTube.
I'm going to go back and see.
I'm not going to bring it up on my screen because then I'll be staring at it. I'll just totally lose track of TWiV the rest of the episode.
I watch a lot of things on YouTube, but I don't watch plaque assays. I will now. I will.
This paper does an interesting twist. They take several ways of looking at the question: is there more than one virus in a plaque? Yeah. The first way, they have a set of 10 polioviruses, which are basically barcoded. Okay, you could use a barcode reader to distinguish them. No, just kidding. They have different sequences in them which don't affect viral replication. They're not lethal, but they could be identified. You identify by hybridization or PCR, whatever you want to do. And they simply mix them together and infect HeLa cells. They do a plaque assay, and so you get isolated plaques, on a plate, so you can pick them cleanly. And then they pick plaques, they amplify them, and they ask what's in each plaque.
And you're assuming, other than this experiment, that each plaque represents a single viral hit to begin with.
So this is based on the one-hit growth curve. However, 2 viruses could land on one part of the monolayer.
I did read this paper.
Right? It could, by random chance, they could land in the same place. Or, of course, viruses are well known to stick together. So it can happen.
But wouldn't you get twice as much virus then out of the double hit than out of the single, and the plaques would be bigger in the double hit than in the single hit?
They're not typically uniform, depending on when they initiated infection.
Simultaneously.
Not only that, but you're talking about in the initial infection, you're talking about one cell infected with either 1 or 2 viruses, and I think the amount of virus that you produce has more to do with the capacity of the cell to produce viruses than it does with the input.
Yeah.
So you're
right. The multiplicity of infection matters a lot for the first cell to get infected with one virus. But then after that happens, the immediately surrounding cells in the next round, it's always going to be a high multiplicity infection because that cell just burst out with however many virus it's capable of producing.
Sure.
And so, you're probably getting multiples of that virus going into the next door neighbor cell because 1,000 of them just came out.
Right, often on a plaque assay, you will see the plaques are circles. Sometimes they're superimposed
over,
and you can see that 2 very close cells were infected just by chance. Right?
Sure. Right. So, but your serial dilutions should get you to a point where you've got very few plaques on the plate.
Yeah.
And at that point, if you still have a double, that means something different, right?
Right. That's the point.
Yeah.
But the question here is, if you have a single nicely isolated circle, did that really start with a single virus?
Exactly.
And so they used these barcoded initially, and they came in with a very low multiplicity to get nice isolated plaques.
And the other question, of course, is do all these strains of polio replicate at exactly the same rate?
Yeah, they think they're all pretty comparable. That was one of the issues they wanted. And they do address that and make sure that the infection was even and so forth.
Is there a way to slow down one of them?
Well, you could.
And then you can tell right away whether it's 2 or 1.
Well, the thing is here, Dickson, what we're going to see at the end of this is that you can have more than one virus in a plaque. But it doesn't mean that you need both of them.
Yeah, but it's very upsetting to virologists that always.
I'm not upset.
No, it turns out also, you'll see, Dickson, at the end, it doesn't really change things for us.
No, it doesn't. Because if we
this is kind of like Einstein's revolution in physics changed things a lot at the really, really, really tiny scale.
No, I know that
if you're pitching a baseball, Newton still works beautifully.
I got it, and this doesn't happen very often in every situation. So.
Well, this is also one virus, so it would be interesting to look at some of it. But anyway, when they do this experiment with 10 barcoded polioviruses mixed together, You do a plaque assay, pick plaques, and then see what's in them. 123 plaques they looked at. 6 had more than one virus.
You're right.
And 117 had a single virus. So 5% contained more than one. It does not mean that you needed both of them, right?
No.
Right.
I mean, it could, but this experiment doesn't tell you that. And I would say because the dose response is still linear, you just need one to start an infection, right? But it's interesting that there are more than one. And they show that, they do another kind of assay. Instead of using these barcoded viruses, they use 2 different poliovirus mutants. What's the matter?
No, I just had another thought, and that is that when you...
You're the only person who could wave to me and get my attention.
This is true. This is all true. I'm waving to Kathy, actually, because I want the answer from her. So when you make your dilutions in the, let's say, PBS, and then you use the exact same volume for all of the culture plates to start cultures properly, why don't you keep it at, like, say, 4 degrees C first for maybe an hour to allow all the viral particles to settle down and all have an equal chance of hitting the receptors, and then raise the temperature so that you start out the same?
Dickson, you're brilliant.
Yeah, that's
brilliant.
That's
People do that.
People do that. They can synchronize.
Okay.
People do that.
All right.
It turns out, Dickson, that by
really.
absorbing for a short time, like an hour, and then diluting, you're basically synchronizing the infection. But in biochemical experiments, you can absorb at low temperature. There will be no entry until you raise the temperature. That's absolutely right.
Okay, fine.
So I have a picky technical question about this that I just have to ask to get from you guys. They say that they are putting down 10 to the 6 cells for the infection, and they're using a 10-centimeter dish.
Big.
For me, they want to get the plaques well separated. But I'm used to a 10-centimeter dish having on the order of 10 to the 7th, or 2 times 10 to the 7th cells on it.
You're right.
So, so 10 to the 6 cells on a 10-centimeter dish seems like it's seated really thin.
You're right.
And they're looking 2 days later. So that didn't compute to me. Does it not compute to you either?
It should be 10 million. You're absolutely
right. My cheat sheet 4x6 card says HeLa cells, 100 millimeter plate, 1 times 10 to the 7th cells.
Okay.
Yep.
Another question is, since not all the cells are infected with the virus, and the virus starts to eat away at a particular cell and then spread out linearly, what about the division of the cells to fill in the gaps after the virus is over here someplace? Could you fill those plaques back in with the same cells that survived this?
No.
And because
but you could fill in some of the spaces and provide more host cells for the virus to infect if you had a subconfluent plate like Rich was just talking about.
Okay.
Right. That's the only way I can sort of make this make sense to me.
Okay.
And if you have a virus that for some reason infects inefficiently or something like a lysogenic phage, then you will get a turbid plaque with some cells inside it.
Which is the name of your website.
Turbid
Which is the name
plaque.
of my website, yes.
By the way, Alan Dove here, when he was here, he did plaque assays.
He did.
Oh, I certainly did.
So all of us.
Everybody else did too.
Except you did a plaque assay.
But I'm working towards it. Maybe I'll
You could come in the lab. Would you like to come in and learn?
Yeah, I might.
It's
Dickson,
easy
you'll
and
never
fun.
leave. Once you've done a plaque
It's easy and
assay,
fun.
You just can't stop.
Yeah.
We have a lot of plaque assays ongoing these days.
Yeah, I noticed.
It's so much fun to develop them. It's like back in the old days when you would shoot photographs on black and white and you'd put the print in the developer and a picture would appear.
Sure.
Plaque assays, you rinse off the crystal violet and there are your plaques.
For me, it was
really
immunoelectrophoresis. I used to love to do that.
One of my earliest experiences, I was doing a time course of virus infection when I was a PhD student.
Yeah.
And I did a 24-hour time course. So I stayed up all night and took time points. Did a plaque assay. It was influenza virus.
Yeah.
I forgot to add the inoculum.
Oh.
Didn't add any.
We did 2-hour samples for 48 hours at Rockefeller once.
I said, never mind. I don't need to do a 24-hour time course.
I had to write my name on a piece of paper every 2 hours to make sure I knew who I was.
By the way, Kathy, we have to put, for polio anyway, we have to put albumin in the PBS. Otherwise, the virus sticks to the plastic tubes and we get irreproducible dilutions. Remember that, Alan?
Oh.
Wow.
Oh.
Yeah,
We used to put albumin in the PBS because we went through a period of time where we did all our dilutions at room temperature, and we had some experiments where we did the serial dilutions and they would sit around for a long time before they went on the cells, and we noticed, this took some figuring out, but we noticed over time that the virus that was diluted way back would die over time in the PBS. And so, but if we stuck some albumin in it, it would stabilize it, and everything would be fine.
We used to use serum, but then we found that some batches had inhibitors in it. So we just...
I'm sure you meant lose their infectivity rather than die.
Oh, you're listening, Dickson. Good.
And Vincent, I still set up my dilutions in glass tubes. I'm a dinosaur, so maybe it wouldn't be an issue.
With the little metal caps?
Yep. I can do it way faster than with a pipette man and yellow tips.
I want you to know that for the vast majority of my career, not only did I use glass tubes with the little stainless steel caps, but we used glass pipettes that we washed.
Yeah, we did.
That's what we used.
I did that in the beginning. Do you reuse the glass tube, Kathy, or do you dispose of them?
Oh yeah, no, they're washed and reused.
Because there are disposable ones.
We had to switch to disposable ones when we started using albumin, because you autoclave those suckers, and the albumin just gums up the insides, and it's hopeless. Take you forever to wash them.
We could tell plaque assay stories for hours.
Yes.
Yeah.
And we're
only up to figure 2 in the paper.
That's all right.
I had an assay
when
once.
I visualize myself in the laboratory doing something, it's usually standing over a light box looking at plaque assay.
Look, Harold Varmus
waxed
waxed poetic at our 400th.
About what?
Yes.
About doing assays, developing an assay.
Yeah.
The whole thing is about an assay.
Yeah,
of course.
Yep.
I had an assay once where I was taking female trichinella worms and putting them in a 96-well plate with growth medium, and then they would shed their newborn larvae over time. And I loved doing that. That was just fantastic.
I can see that, absolutely.
It's raw data. It's absolutely fantastic. Immune worms, non-immune worms, that sort of thing. So, I love anything that's a process and explicative also. You get to explain something afterwards that you didn't know before.
And just think of all the people who are doing a plaque assay as they're listening right now.
You found that from your mail. That's true.
Yes, many people do.
That's right.
Actually, to me, one of the most fascinating things about plaque assays, and this goes for microbiology in general, but it shows up for me best in a plaque assay, is, of course, we're working on these microorganisms. You can't see them. All of our assays are indirect.
Exactly.
And a plaque assay is even less direct. It's all you're doing is counting plaques. And you sit there and you look at these plaque assays and count the plaques and make up these fantastic stories in your head with all these pictures.
Exactly.
But all you're looking at is numbers.
Exactly.
Yeah,
that's the whole deal.
Well, back to the paper.
Indeed.
So
forget the paper. Let's go to the email.
They validate this using a separate assay where they have 2 phenotypically marked viruses, right? One is drug resistance, one is temperature sensitive. And they can do the same co-infection and ask in a plaque which 2 phenotypes are present. And they basically get similar results, 5 to 7%.
Some of the plaques actually get both the drug resistance and the temperature resistance, which shows that some of those plaques were, in fact, dual infected.
10 out of 100.
Okay, so now I have a bone to pick with this.
Yeah.
I mean, it's clear that the conclusion is valid. They're double infected. But I'm looking at the figure here that has plaques on both dishes, 32 degrees plus drug, 39 degrees minus drug. And the assumption is that the 39 degrees minus drug are the guys that are not temperature sensitive and drug sensitive, and the guys at 32 degrees are the guys that are
the other side,
temperature sensitive and drug resistant.
Right.
But you don't know that just from looking at the plaques. They could be, as a matter of fact, they could even be recombinants. Okay, they aren't necessarily different viruses. So, class, how do you prove it? How
would you mark them?
Well, I think the previous experiment was the way to do it.
But with this experiment, how do you prove that those viruses are what you think they are?
You want to pick them?
Yeah, absolutely.
Okay.
That's the answer every time.
And
you pick the plaques and test them individually and make sure they're what you think they are.
Okay. 10 out of 138, 7% had more than one virus. All right. Now, why? They look under the electron microscope and they see
Ollie.
Single particles, a lot of single polio particles, but also some aggregates.
Do they?
And that would explain getting multiple infected.
My goodness.
Viruses can stick together. We know this.
Yeah.
And in fact, they do an experiment using a green and a red polio, and they incubate them at low pH buffer which induces aggregation. They show by EM that there are some method that, what is it?
They used flow cytometry.
Because these are fluorescently labeled.
Look at aggregates.
They're aggregating, and the frequency of co-infection goes up when you artificially increase the aggregation by incubating at low pH. Okay, so
Vincent and
yes,
Kathy, with polio and adenovirus, when you do a plaque assay, do you make any attempt before you do your serial dilutions to disaggregate the virus?
No.
No.
Because for both polyoma and vaccinia, the two that I have the most experience with,
You have to.
aggregation is a known problem.
Sure.
And I learned polyoma first, and the technique that I learned there was sonication. They would take the lysates and stick them in a sonicating water bath for ever.
Really?
Is that an envelope virus problem?
No, because polyoma
or is it due to
wasn't envelope.
Not an envelope. Okay.
And so it could be sticking to debris or something like that. In vaccinia, it's an envelope problem, and in Dickson, I've always sort of imagined that as the envelopes being sticky.
Yeah.
And when I first started working on it, the technique was to actually trypsinize them for a limited period of time. And you could see if you did a plaque assay on untrypsinized virus and compared it to a plaque assay with trypsinized virus, when you trypsinize it, the titer would go up significantly. So that was a big deal. But I didn't like trypsinizing them because, first of all, it was laborious, and second,
you're taking a chance here.
Yeah, so I introduced sonicating them, and we used to sonicate.
I think a good experiment here would have been to sonicate polio and look under EM and see if you could get single particles and if that gets rid of the dual infection. Right?
I was thinking the same thing as I was reading it. Can you do something to disrupt these aggregates and then confirm the opposite?
Yeah.
So let me get this straight. The viruses that aggregate together infect together?
Yes.
That's it.
Apparently.
Rich,
Yes.
then did you have to sonicate a stock once, or you sonicated it just before the plaque assay? If you did that, does that mean that you would sonicate just before you use that stock in every subsequent experiment?
Okay, so this blew my mind. Okay, first of all, this became kind of ritual, and you do the experiment a couple of times to show that the sonication actually has some sort of an effect, and it wasn't totally reproducible, but you feel like, well, I got to do it, because it could be, and it's not hurting anything. And we used to. First of all, we worked with lysates. We didn't work with purified virus. So we usually worked with lysates. So that means you have, for the Caleb's out there, that means you have an infected culture of cells, and the virus in vaccinia stays cell associated. So you could spin down the cells and then resuspend them in a little bit of buffer to that step concentrates it, and then freeze and thaw about 3 times, and it breaks all the cells. And then you don't do anything else. You got cell debris and virus sitting there, and you keep that in the deep freeze. And when you go to do a plaque assay, you thaw that, and every time we would sonicate right before we did the serial dilution. So you thaw the virus, you sonicate it. We used a cup horn, right, which is a really high power water sonicator. We'd sonicate it in the cup horn and then do the serial dilution and then freeze it. And we did that every time. Now, the weird thing is it's not weird, but it's cool. We used exactly the same technique on solutions of DNA to smash DNA down to 300 nucleotide bits to clone for shotgun sequencing. Okay? So that amount of sonic power will absolutely shatter DNA. But you could take vaccinia virus and do this over and over and over and over again for years, and it was fine. The titer was absolutely stable.
So, Rich, when you were doing your trypsin experiments, did you ever think of adding a trypsin inhibitor as a way of stopping the reaction so you could control
usually what would happen is what you do is you could show, and we actually did this experiment, that you could over trypsinize something. So there was an optimum. It was like 15 minutes at 37 degrees with a certain concentration of trypsin, and then you dilute it about 3-fold with media that had serum.
Oh, I see.
And the serum
has
distracts the trypsin.
The serum has a trypsin inhibitor in it.
Okay, fine.
All right. Last experiment.
Wait.
Go ahead.
Rich didn't quite answer my question. So when you then take that, so now you've frozen it down in aliquots. If you take an aliquot and you're going to do an experiment with it, not a plaque assay, some experiment, Do you sonicate it again, or you just trust that it's going to be able to infect at the multiplicity you design based on the titer that you determined when it had been just sonicated?
You sonicate it again. The assumption is that when you freeze the virus, it reaggregates.
Okay, so you sonicate before every time you thaw a virus not to do anything with it.
That's right. That's right.
Wow.
And it's like sort of my thinking in the long run, as I say, we only did the quantitative experiment a few times. And sometimes it made a significant difference, and other times it didn't make that significant a difference. But it never hurt. Okay, so we figured, you're better off doing it. And I can say that over time after time, you'd retiter the same sample and the titer would come out the same. The vaccinia is very stable at minus 80. So you'd get the same. Not only that, but if you wound up doing infections at different multiplicities for different reasons and looked at the monolayer after the infection, the fraction of cells that were infected made sense with the multiplicity that you were using. So I'm confident that protocol of sonicating every time was a perfectly reasonable way to do it. That's the way we did it. Yet I want another footnote here. Listen to this conversation, okay? This is science, all right? All of these nerds sitting around getting all excited about plaque acid.
You're absolutely right.
We've gone way out in space.
Even a non-virologist is getting excited
because everybody wants to know, how did you do it? Did you this way? Did you?
Yeah, that's right.
It's great.
Yeah.
Well, that's the nuts and bolts of science. That's right.
Yeah, this is it.
And this is what, if you're outside of science, you can't quite get unless you get into it.
That's why we do this.
And the way you learn is you go in a lab and you do something that doesn't work, and someone says, you numbskull, this is the way you do it.
You dopo.
You have to sonicate it.
Listen, I remember the time I did my first sequencing gel, and I did everything perfect except for just one little step. I did it perfectly except for one little step.
What was that?
I forgot to boil the samples.
Yeah, well, I forgot to add the virus, it happens.
It came down and it looked
so there's
horrible.
one other little technical variable that you can add. Some people for their overlays use methyl cellulose. And that happened, my lab sort of morphed over into that over time. So that's methyl cellulose is sloppy, but it's at 4 degrees, it's kind of this sloppy mixture, but if you stick it on the plaque assays and put it at 37, it gets not quite so sloppy. And it's solid enough so that it inhibits the diffusion of the virus in the plaques, but you can just suck it off. You don't have to dig off the monolayer or anything like that. So it's a little easier to work with. And you can keep a bottle of it you don't have to make it up every time. You can keep a bottle of methyl cellulose in media if you want in the refrigerator, and then when you're ready to do a plaque assay, you just heat it up to 37 and stick it on the cells.
I always kind of liked flipping the little agar bits off.
That's great. So Saul Silverstein, who did herpes here, used to use methyl cellulose. We tried it. It didn't work with polio, but now and then, if you bump into the incubator, he would then develop the plaques later, and they'd all look like little comets.
Yep.
Right? Because you've moved the overlay, moved the virus.
Yeah. It can happen even with agarose.
Yes, I'm sure. The final experiment here, what they do is they take these drug-resistant, temperature-sensitive mutants, and then they keep one untreated. Then they put another in a background of a RNA polymerase, which makes fewer errors than wild type, and then they take wild type and they mutagenize it with ribavirin, which is a mutagen. So they have 3 populations now, lower than wild type mutation frequency, wild type, and then more than wild type mutation frequency. And the idea is under these conditions, when you introduce potentially lethal mutations, you get recombination and complementation. So you need to have more than one virus to form a plaque, right?
Right.
So they figure out the co-infection frequencies in the same ways that we've been talking about. So the wild type virus, 7% co-infected plaques. If you have the polymerase that is making fewer errors than wild type, 4.4% co-infection, so a little less. And then treat with the mutagen, 17% co-infection. So the idea here is that if you need to complement to get a plaque, you need more than one virus, which makes sense. And I was talking to Steve Goff about this before, and he was saying how with SV40 they've done this over and over. They'll take two completely defective mutants, and you can show that you need to get both of them in a plaque. And in fact, that's a two-hit curve, which is parabolic, which is very clear and very different from the kind you get with this. Now, the authors, from all of this, they conclude these data do not conform with the one-hit model. All right, so I would disagree with that, because just finding two different viruses in a plaque doesn't mean you needed both of them. They were there because the virus was sticking together. And the one-hit curve of Dulbecco and Vogt still holds, right?
Yeah, I agree.
But
I agree.
Now, I agree that if you mutagenize, you might need more than one. That's perfectly reasonable.
Well, so there are two questions to me in the one-hit model. One is, do you need more than one virus to infect? And the answer with polio is clearly no. But the second is, if you have more than one virus infecting, that is something that's something that I do consider inconsistent with the one-hit model.
No, because it's not required, though.
Well, it's not required, but
that's what the one-hit model means.
But you're getting sometimes simultaneous infections, which tells you that sometimes one plaque did not come from one virus.
Yeah. Yeah.
So it's relevant if you're plaque purifying and you want to be lazy and only do one plaque purification.
Is that what you used to do? Is that what you used to do?
No. I never did that. Plaque purifying.
You do run the risk with plaque purification that you fix some rare random mutation.
Yes.
Mm-hmm. And it becomes an issue, for instance, in your ratchet figure, Vincent, that you use in your lectures, where if you do it 30 times, it's a real problem. But even at 3, you might run into that.
Yeah.
So.
Yes.
Yeah, I agree. So you should plaque purify 3 times. I think that would get around this sticking problem.
Right. So the one-hit model is still valid in the sense that certainly for this virus, you only need one hit to infect the cell. But I think what they're saying is that you sometimes do get more than one hit, and we need to bear that in mind when we're interpreting results that involve plaques.
Yeah.
Okay, so I have 2 points I want to make.
All right, let's get sassy. Get sassy.
Yeah. One is, if you think about the title, the plaques formed by mutagenized viral populations have elevated co-infection frequencies. Okay, so that's basically looking at the results of Figure 5, which I think might be prone to misinterpretation if people don't really think about the experiments carefully. But what about the following? If you just consider that in your highly mutagenized virus stock, there's going to be some particles that have RNA in them, but they're not going to be able to replicate, but they still get in, then in a way, the effective multiplicity of that stock, of that highly mutagenized stock, at least with respect to the RNA, is maybe higher than what you're putting in if you're trying to put in, for instance, equal multiplicities with a wild-type virus. Did you follow that? Did I make that? Anybody with me?
I think I followed. Yeah. I mean, basically, it's not like the co-infection. Mathematically, the co-infection frequencies are going up, but that's only because when you highly mutagenize a virus population, you're killing a lot of these dudes.
Right. And I guess what I'm trying to say is that in this kind of experiment that you might be putting in a whole lot more of your highly mutagenized stock than you are of the wild type. And so they might be landing. Anyway, so
you have
I
to,
guess,
right? You would have to put in more to rescue. You'd have to get co-infections to rescue. And the aggregation might not account for that. You have to actually get co-infection of cells with 2 different viruses, single viruses, so you'd need to have a higher MOI to do that. Right.
So I guess what I'm trying to say, I may have kind of lost the whole thing, but if you're normalizing what you're putting in just by normalizing by titer, your highly mutagenized stock has effectively a higher MOI of particles with RNA in them. So a higher particle to PFU ratio. And so that might, so if you do have a tenfold higher mutagenesis, more highly mutagenized, you get more detectable co-infections in a way that almost just seems like, well, duh.
Yeah, of course.
Okay.
I
So
agree.
I guess that relates to the title.
Yeah.
Okay. And then the second thing, it kind of comes back to, I think it was Dickson that asked about it, is this really going to change how we do things? And with respect to the one-hit kinetics. And I don't know what you meant, Rich, when you said that your plaque assays were highly stable or your titers were highly stable. But if the variance from one time you titer a virus to the next is maybe twofold different, then that's already 100%, where they're talking about, 50% of the time you're getting these chimeric plaques. So I guess
No, they didn't find.
in other words,
50%.
the variance in the plaque assay is larger than this effect of co-infection.
Right. And
I-
I always stressed in lab and in classes that any two plaque assays that are within twofold of each other are about as
It's the
good
same
as you're
result.
going to get.
As good as you can get. Yeah. Okay.
Absolutely.
So that's because then they go into this thing about the you're only seeing 1/3 of the events if you're looking at two different phenotypes. You could, if you have virus A and virus B, and if you get a co-infection of A with A and B with B, you don't see those events. You only see the A with B as a co-infection event.
Right.
So you're only seeing 1/3 of your events. So then they say that if they take their 16.7% and they multiply by that factor of 3,
I see.
that's where they get their 50%. And I'm saying that if a plaque assay of a stock varies twofold, that's already 100%. So that was my point.
Yeah. So I think
Sure.
a better way to look at this, Steve Goff said in the old days they would take 2 SV40 mutants. Each on their own will not replicate. No plaques. You co-infect, you get plaques. The dose response curve shows 2-hit kinetics. It's very clear. Right. So, the mutagenesis, you kind of lose that because you can't see how many, like you were saying, Kathy, the 50% is sort of invisible. You have to do math to do it. But with this SV40, you quite clearly show that there's no plaques with either virus. You have every plaque you get when you co-infect is doubly infected because the curve, the dose response is parabolic. And so that proves that you need to have co-infections when you have mutated viruses. I think that would be the experiment to do.
So I have 2 comments.
Yes, sir.
One is that I have a suggested for a title for this, and that is 2 virus particles walk into a cell. The second comment I have is when one virus particle infects the cell, you get X amount of virus and you get the plaque growing at a certain rate. When 2 virus particles go into a cell, do you get twice the rate?
No, as Rich said earlier.
Okay, no, that's
you asked him.
I did, but I didn't really.
He said it was not going to make that much difference.
Grasp why is that? No, I want to know why that is.
No, look, Dickson.
You get half the number of each of the 2?
No. It takes one woman 9 months to have one baby, no matter how many men you put on the job.
Ooh.
And there's something similar going on here, where you've got the infection cycle for the virus is going to take a certain amount of time, whether you put one virus into one cell or 10 viruses into one cell.
Yeah, but the quantity of virus is what I'm asking
I
about.
think you're going to come out very close to the same.
Yeah, you come out very close. As a matter of fact,
meaning that each of
as a matter
the 2
of fact,
viruses replicates at half the amount to equal
Not
the equivalent of one
necessarily.
virus. Well, wait a
I
minute,
think
that's just what you said, though.
my experience
No.
is that a cell has a certain capacity to produce virus.
Yeah, I would assume that, too. So for the double hit, you get half the number of each of the 2 viruses to equals what
I
one
see.
virus would.
Assuming that they are replicated at exactly equal rates,
Because
then
the plaque
yes.
is the same size.
Yes.
Right. We also
So that means you can't tell by plaque size which one is a double and which one's a single. That was my point.
right. Thank
Thank
that's
you.
the point. That's why
No,
they
that's-
had to assay these this way.
That's why they had to do this paper.
The light just came through the window.
Right.
Okay.
Right. But at the same time, this doesn't change anything about the Poisson distribution or the relationship between number of plaques and number of viable virus particles in the original sample. I mean, as Rich said,
No, that's true. But
this is within tolerance for the assay.
so you would though accept the fact that the cell that got a double hit produced half the number of each of the 2 viruses to equal what one single particle would have.
Assuming that they replicated at the same rate once they were in the cell.
Exactly.
I mean, if one of them was
theoretically.
mutated,
So you can test that by then trying to titer out the double hit plaques from the single hit plaques for
Yeah,
numbers
and
of
that's
viruses.
exactly the question that Rich asked the
Okay.
class
Okay.
earlier. How would you test
So
that?
I'm a student. I'm in the front row. I'm waving my hand like crazy. And the guy says, Horshack, you've asked every goddamn question in this class. Shut up.
Horshack?
You remember Horshack and Welcome Back, Kotter?
I
Back
never-
Cotter.
I know,
never was.
Horshack was always,
Cotter.
but Mr. Carter, I want to ask a question about,
Yes.
that was Horshack's
So,
voice talking like this.
Yes, Rich.
To address your question in general, I know from experience that if I take, a bunch of dishes of cells and infect them with varying multiplicities of at least the virus that I worked on, anywhere from 0.01 up to 10, and let the infection go until all of the cells have been infected and they've all released virus, and then I do plaque titrations on that and calculate the amount of virus that came out of each cell. It's the same, whether the multiplicity was low or the multiplicity high.
One other question, of course, does the cell cycle of the host cell matter as to when the virus enters it, as to how much virus you'll get back?
Really great question. There are some viruses that are, at least to some extent, cell cycle dependent, and others that aren't. As a matter of fact,
Is this one?
Polio doesn't. You could take the
polio doesn't care,
you could
right?
take the
Doesn't
nucleus
care.
out and it would replicate.
Really?
But many DNA viruses need the cells to be
So
divided.
you could give a whole cell culture scene, allow all of them to come to metaphase state. And
Polio's okay. Polio's okay.
That's incredible.
Yeah. That's why it does well in neurons, I guess.
I
right?
love that. No, that's fantastic.
Yeah. Okay. Are we good?
We're good.
All right. I just want to do 4 emails.
By the way, this is as much as I've ever talked about viruses.
I think you've
just
talked
want you to
before.
know.
You've talked before. I'm glad I engaged you. We engaged.
This crew
No,
really gets off on plaque acids.
Very-
That's for sure.
Yes.
Very
I'll tell
no.
you.
This is the guts of research. I agree with you, Rich. This is the way it's at. If you understand this, you can understand everybody's experiments.
Well, if you can just feel
Yeah.
the enthusiasm.
I feel the
I
tension.
mean, this is why people get hooked on this stuff.
I hear you.
Okay,
I hear
it's
you.
because you have these conversations.
So here, I would like to hear someone in parasitology do the same thing for some assay they do,
Well, it's
right?
they're not
The
being
assays are a
funded
lot
at your level.
harder.
Or any other field.
It's tougher.
Immunology has a lot of cool cell
I think
assays,
malaria
right?
and toxo have a lot of people.
Tetramer staining. Tell us all about
Yeah, we can do that. Right.
So that's very few people do these kinds of shows where they
That's
get
right.
down
That's-
into
you're
it,
right.
right? And I
You
think
are
people
right.
like to hear it. I'd love to hear
Yeah.
some other fields.
Yeah.
Well,
Yeah.
that's why I did micro and parasitism, but
Yeah.
No, wait a minute. I was at a meeting in Africa once. My only meeting is in Africa. I went to Ilred, and somebody had a gel, and he didn't know what it meant.
gel,
A gel. He had a gel, and he
protein
said,
gel.
if anybody here can stay after the break, I would like to show a
Yeah,
picture
that
of this
happens.
gel.
That happens.
And when we came back after drinking our Tusker beer for an hour, they were still discussing the meaning of this gel.
You went off to drink beer?
Oh, you betcha we did. Tusker
What is
beer.
Tusker?
That's the brand of beer in Kenya.
Sounds good. Is it made of tusks?
No, it's made of elephant urine. No, it's not made of elephant urine either. It's made from beer. It's like,
Tusker.
like you would drink.
Oh, I'm looking it up now.
But the thing that I was really amazed at was how intense the arguments were about the meaning of this gel.
Yes, meetings.
It's
That's why
fantastic.
meetings are cool.
It was great. And the Gordon conference was filled with stuff like that. I loved it.
Yeah. Well, Gordons tend to be smaller, so you can have more animated.
Yeah, that's right.
Discussions.
And into the evening and in the
yeah.
early morning.
There's nowhere else to go usually, right?
That's
You're
right.
stuck in New Hampshire or
You
something.
are. You're stuck in New Hampshire. Unless you like night fishing.
Andrea writes, suggestion, perhaps you could pick a random email out of the emails that you receive in a specific amount of time, say 2 weeks. This way, people that listen to the podcast early and send in an email have the same chances as those that listen later. I am assuming, of course, that you want to get the contest over as early as possible so that you can continue to clean out your office of all the extra books. I think that's a good idea.
Not bad.
And Andrea is from Seattle, and she puts a little umbrella with raindrops
A
next
little
to it.
emoji of a rainstorm.
Yes.
So my problem, being some marginally digitally challenged, is that how do you select at random an email? Okay, I mean, I can
Could
I can
number
reach
them.
into
You
a
could
basket,
number
pick
them.
out a number if I got a bunch of pieces of paper in it, but I got a bunch of emails. How do I pick one at random?
Well, actually, someone else has an email which I didn't put on. He said there are random number generators that you can find online, and you can just number your emails and use that to pick one.
Yes.
Okay.
We could do that. So we could say after 2 weeks, I'm going to run this thing and pick out an email. I think that's a good idea.
That's a good idea. That's the way Car Talk did it.
Is that right?
Yeah. Send in the postcards and they get a gazillion postcards.
Right.
And after a certain period of time, they just pick one out of a jar.
Yeah. The person who sent this in said on one of the podcasts he listens to, they give things away and they use random generators.
A random email.
Yeah. Rich, can you take the next one, please?
Ken writes, Vincent and crew, when I heard you were giving away books, I assumed they'd be gone long before I could type your address on my email. If you have not yet, please consider this submission. I've been listening for a little over a year since my friend L'Oreal introduced me while we were talking about her research on AAV. She met you in Portland, and I remember hearing about your trip on TWiV. I was also pleased to hear you read her email detailing the capsid proteins on which her thesis was written. I'm a software engineer and my knowledge of viruses is very limited, but I'm a huge fan of science and learn as much as I can find time for. That's why I love listening to TWiV. I have to pause often and look up terms and papers. It keeps me on my toes. Keep up the good work. P.S. It's cloudy, windy, and about 10 degrees C here in Portland today with a lot of rain in the forecast. Ken.
Love it. Next one's from Alexandra, who also is writing about the book, but she writes, I just got into the wonderful world of viruses 3 months ago, and you have definitely infected me with great fascination. And Alexandra is a virology lab research assistant at the University of Puerto Rico.
Cool.
Love hearing from all the people. Alan.
Donna writes, Dear TWiV crew, I heard on TWiV 428 that you'd not yet received email number 27 for the Emerging Infections book. Would be great to add this to my collection. Even if I'm not the winner, I want to thank you for the TWiV podcasts. I've been listening since the Joan Stites interview. My graduate work was in virology, VSV inhibition of splicing in Jack Keen's lab in the 1980s. I meandered into other topics for postdocs and beyond, but I'm a lecturer at RPI, non-tenure track, 10-plus years, where I teach a variety of molecular biology-related courses. I was excited when virology was added to my teaching duties in 2015. This led me to your podcast via Viral Zone. I especially enjoyed your discussion of VSV ultraviolet inactivation, as that was a technique I used extensively in my graduate work. Thanks so much for your podcasts. And Donna is a lecturer at RPI and Biological Sciences.
Yeah, we just did the VSV paper in a discussion today. And I showed Andy Ball's letter that they loved it. They just loved it.
I think it's cool that she found TWiV from
Viral Zone. Yeah,
Viral Zone.
Yes, they very nicely have right on the front page a link to TWiV.
Yeah, I'm just looking at it.
Which has been there for many years, which is great. Kathy.
I have Neil. Yes, Neil writes, Sparrow urgent care physician Dr. Song Yu says, although it feels similar to the flu, it's actually a virus. Some other good quotes from the MD in this article. Maybe Kathy can try to do some in-state education. Wow. So I looked at the article, and it could be that he wasn't quoted right.
Because what he says is the same thing.
It can start as early as September to late April, and sometimes. And right now, I'd say it's peak season right now. And then this line.
It's the second paragraph.
Although, it feels similar to the flu. It's actually a virus. He might have said it's actually another virus, and the reporter didn't pick up on that.
Yeah, maybe. It's not a quote. It's a paraphrasing.
Yeah. So I'm hoping that Dr. Yu got an education about viruses and other things, and influenza is a virus.
Maybe speak to Alicia Bergio or Berglo, who wrote this this article.
Yeah.
Oh, this is a virus going around mid-Michigan.
Right. Right. So, and then P.S. from Neil, to add to the other podcast list, Freakonomics, especially my listener pick 3 episodes called Bad Medicine. And he gives the link to those: Bad Medicine Part 1, Part 2, and Part 3. So check out Freakonomics podcast.
Right. And finally, Dickson, one more.
Sid writes, Hi, TWiV team. It's 6 degrees C in St. Louis, and I'd love to get a copy of Emerging Infectious Disease book. I'm a PhD candidate at WashU studying RNA phages and viral dark matter. I've been listening to your podcast for about 8 years now, primarily TWiV and TWiM, and I think it's great. Thanks, Sid.
8 years.
Yeah, I think he's been listening to it since before it started. Right?
We're about 8 years old, right?
Are we 8 years old?
2004, 2005.
2008. That's longer than 8 years, right?
Okay.
Yeah.
Well, time flies when you're having fun.
It do.
All right. The next one's very long. We're going to take it next time. And let's do some picks of the week. Rich Condit, you have been gone a long time. Let's take you first.
I have. I went out to lunch with an Austin TWiV listener. And I'm available for these.
It's not
for these
Neva. It's not Neva, right?
No, it wasn't Neva. It was Tom.
Cool.
He's written in a few times, and he turned me on to this puzzle that you can do on your phone called Hook, appropriately named. Have you guys tried to play this?
Nope, I didn't try it.
Nope.
No.
Okay.
Nope.
I haven't played this one.
It costs,
but
the link that I put here gives links to both the Android and the iPhone app. Mm-hmm. It's available for both and other platforms as well. It costs 99 cents. It's worth it. I don't even play the sample, because part of the fun here is figuring out even what the puzzle is about. Just no instructions, forget about it. Just go in and play the puzzle. I probably shouldn't even tell you, but I'm gonna. There are 50 levels. Okay, and
that
it
was
probably
a
only
spoiler
takes
alert.
if you get heavy into it, it takes less than an hour to get through the whole thing. But it's a hoot. It's really good.
Cool.
I've played a few like this. This looks like fun.
And it's a lot of fun.
Hook.
Cool.
Did you have a nice lunch?
Yeah, it was great. We went to one of the oldest classic restaurants in Austin, and now I'm blanking on the name of it, but it was he told me that it was originally the last gas station in Austin as you left town to the north. And it became a speakeasy, and they got the first liquor license, and I had, at his recommendation, because I had to do it, I had chicken fried steak, which I'd never had before.
It was good, I bet.
We did the whole thing, I'll tell you. It was great fun.
What is chicken fried steak?
It is like
kind of what it sounds like.
Like a cube steak. Okay,
Yeah.
so it's some sort of inedible piece of meat.
That's right.
That is pulverized
Correct.
to make it edible.
Correct.
And then thickly battered and deep fried.
That's
and it
right.
comes
Why is
it comes with this gravy like they have in biscuits and gravy over the top. It's this sort
It's
of
a very Texas.
gray stuff.
Why is it called chicken fried?
Because you fry
That's
it
how
with
you
fried
fry chicken the same way.
Okay.
You put it in batter. You wouldn't cube the chicken, but you'd put it in batter and then deep fry it.
Not sure Blue Apron has that recipe.
Probably not.
No, they wouldn't do that.
We have quite a few listeners in Austin, Rich.
Yeah,
Could probably have a lot of lunches.
I'll do lunch dates, and we'll put all those together, and then we'll have something.
Here in New York, Dixon and I could go to lunch.
Oh yeah,
We can do that.
Going to lunch every day for years.
We could do that.
Alan, what do you have?
I have what I thought was just a really, really good article. This is from The Guardian. It's a love story. It's about a couple who are donating their insect collection to was it University of Arizona State University.
Mm-hmm. It's in Tempe.
They have been collecting insects all their lives together, all their adult lives together as a couple. They met through entomology. He was a teaching assistant, and she was taking the entomology class that he was teaching, and she fell in love with insects and him. And then they've spent their married lives together, traveling around the world and collecting insects, and they have this enormous collection that they've stored in their home, and it's all properly stored and cataloged. And they sit in the evenings with TV on, sorting and cataloging the insects that they've gotten on their most recent trip. So it's just a really neat story about how these two got to this point. And they're now in their 80s. And so they're deciding.
They've been married for over 60 years.
They've been married for over 60 years and collecting insects for over 60 years all over the world. And they've got
nerd love. It's wonderful.
It's nerd love. It's absolutely perfect. They're completely adorable. And they've got stuff in this collection that that these samples are not quite unique, but extremely rare. And so this collection's actually valued at somewhere around $10 million. And it's now going to vastly increase the insect collection holdings at Arizona State. And just really cool.
Wow. It's impressive.
Yes.
$10 million.
Indeed.
And are they going to stop collecting, or are they going to start?
No, they're still collecting, but they say they're cutting their schedule back a little bit now that he's 83 and she's 89.
Yeah, it is a really cool story. I sent it to several people who also enjoyed it.
Yes.
Wow, it's amazing. The house is full of drawers.
Yeah. And mothballs. I'm sure it's got mothballs in there too, because these are very
it's got
susceptible.
Right.
Well, I have a nice poliovirus collection. If anyone,
you've got a whole wall over here.
Dickson, what do you have?
I have an update on the way to look at things in outer space. Obviously, this is a Hubble upgrade, and it shows before and after the upgrade. And there are 10 of these shots, and it's just incredible the amount of detail that we can now see. In things that we could already see detail in and thought were incredible, they're now super incredible. So I just thought you'd enjoy seeing the updates as to how we spend our money going into outer space and doing ophthalmology on the Hubble.
Nice. Yeah.
I mean, look at the Horsehead Nebula.
Yeah, it's amazing.
That's just
really cool.
Incredible.
Would you like to take a trip there, Dick?
I would love to. I mean, if I could and come back. You asked me, will I be able to come back?
Sorry, Dickson.
I have to tell people what I've seen.
Come back.
No, I've got to.
You could Skype.
I don't know. The plans are a little nebulous for that.
Kathy, what do you have?
Well, this is a link to something that AAAS is promoting. Why is science important to you? You can answer this question in a short story, less than 150 words, not very long, and that could get published in Science. And this is part of something they're doing called Force for Science on a website. Submission deadline is today.
Oops.
Sorry, if you're
hurry!
It's too late. I didn't even realize that till just now. Oh, maybe they'll have another one in the future.
But you can go to the Force for Science website and look up other stuff
and check out other people's stories.
Now more than ever, we must demonstrate the importance of science to policymakers and the public. You know, that's what we do here on TWiV.
Only if they'll listen, of course.
Yep.
Well, that's the
whether they'll listen or not, we're going to do it.
Yeah, we're going to do it. The policymakers have to listen. Well, maybe we can talk about this when we do a TWiV before the Science March, right?
Let's talk about plaque assays.
Plaque assays. We could.
We'll get a senator in or a congressman, and we'll talk about
put him to sleep.
Okay,
By the way, the restaurant was Threadgill's.
Threadgill's.
Okay. I recall hearing about that place.
I have 2 New York Times articles which relate to Trump's proposed NIH budget cuts. They're both very well written. The first one that I saw was actually last Sunday in the opinion page on the weekend. It's by an old friend of ours, Michael T. Osterholm from the University of Minnesota.
Oh, heaven.
The real threat to national security, deadly disease. And his argument is that, the president wants to increase the military budget, but we need to also fight infectious diseases. And he goes through several examples of various serious diseases that cause lots of troubles, and we have to keep our guard up and so forth. And I must say, this is the first article in the Times that he's written that I agree with.
Yeah.
Because some of his previous ones, one was, will Ebola become airborne? And another one was about limiting H5N1 research and so forth. And we've talked about all of those, but that's very good. Thank you, Michael.
Yes.
And the second is also in the Times, and it is by Harold Varmus, Why Trump's NIH Cuts Should Worry Us. And this does not try to scare you. So Osterholm's modus is to try and scare people and say, infectious diseases could wipe you all out.
Which is true.
Which is fine, and I think we need to put money in because it could be a problem. But here, he's very level-headed. Of course, he was NIH head for many years, and he knows what's going on. He knows all about Congress and the budget. And he says, probably it won't go through because Congress has been pretty good for supporting biomedical research. It's not just up to the president. But he said it's a mistake to be complacent, because what happens if it's the same as before? That's not good. It should go up. And what if it's a little less? What if $1 billion less? This could be a real problem. And so he goes on to explain about how NIH funding works and so forth and appropriations. It's really well done. Beautiful. He's a great writer.
He is a great writer.
He's also a great speaker, as we know.
He's a great thinker, which is why he's a great writer and speaker.
Yeah, he's a very good thinker. So 2 really good articles on an important thing. You should check them out. And that will do it for TWiV 435. You can find it on iTunes, microbe.tv/TWiV. And also, if you have an iPhone, a smartphone or a tablet, there are all kinds of apps you can use to get podcasts for free. Just subscribe. You should subscribe, by the way. And that way you can get every episode. Even if you don't want to listen to it, just subscribe. Okay? It's a little secret there. Just subscribe. Get them all for free. You can help us out financially. We're beginning to travel together this year to do different things like Washington, ASV, Madison, where are we going? Hamilton, Montana, and other places too. And help us out. Microbe.tv/contribute. All kinds of Patreon, PayPal, etc., etc. We appreciate your support. We will be eternally grateful for your support. And even when Dickson goes to Mars, we'll be grateful.
Everyone will be grateful.
Dickson Despommier is at thelivingliver.org and parasiteswithoutborders.com. Thank you, Dickson.
This was fantastic.
It was the best one ever.
It was very clear.
Good. I'm glad you liked it. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit, formerly a professor at the University of Florida in Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough. It's always a great time.
Alan Dove is at alandove.com. You can also find him on Twitter as Alan Dove. He writes the Turbid Plaque. Thank you, Alan.
Thank you. It's always a pleasure. I'm
Vincent Racaniello. You can find me at virology.ws. I want to thank the sponsor of this show, Blue Apron. The introductory music you hear on TWiV is performed and composed by Ronald Jenkins. You can find all of his work at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. You can find me right next to the wall of polio, right, Dickson?
Yep. Sometimes on it.
You know what? I was going to have the three of us say each other's closing lines because it's April Fool's Day tomorrow. And I forgot to set it up. So I would have said, sure enough, always a great time. Alan could have said, thanks, this was a lot of fun. And
yes.
Rich would say, always a pleasure. But, oh well.
Dickson,
I'll just drop out.
Can you say another TWiV is viral?
Another TWiV is viral.
Yeah, so thoughtful. We have a lot of good titles here. A fishing expedition and a poisson story. Ha ha ha.
Mm-hmm.
Petabytes and plaques. That's pretty cool. A plaque for teamwork. Supernumerary viruses is very cool. I see a red cell and I want it painted plaque. That's good too. All right. Syringe is the new plaque. What does the syringe come
like
from?
Orange.
The blood.
The, black,
the blood? Oh, okay. Yeah. Orange is the new black. Mm. What's red? Plaque and herd all over.
That's a good one.
Placating viruses. And 2 virus particles walk into a cell. Oh, there's so many good ones here. I like 2 virus particles and I like supernumerary viruses.
Right.
I think I see a red cell and I want it painted black is good too. I don't know which one to choose. Who has some favorites? Rich Condit.
I probably 2 virus particles walk into a cell. They're all good.
Dickson, Despommier.
Well, I spoke that, so I guess I should pick it. But,
which one do you like, Kathy?
I'm good with all of them. 2 virus particles walk into a cell is good.
Alan, you okay with that?
I'm fine with that. I'm pretty proud of some of the others. But
yeah, no, you did a great job. It was great. Fantastic.
They can be at an Easter egg.
Yeah, because they're pretty clever.
You should call these outtakes and then publish that along with the show notes.
By the way,
Dickson.
one of the I took notes while I was listening to the TWiV on the airplane the other day.
Yeah.
And I'm not going to go through all of these, but there was that email where the writer, a woman, I think, asked about working in a lab, what it was like.
Mm-hmm.
Yes.
That was just a wonderful email. And in particular, what she was curious about. All right. And we need to do all of that stuff. And in particular, I think it's going to be a while before we get to that, but let's do a bit sometime where we talk about smells.
Yes.
Because I have such strong associations with smells that most people wouldn't be able to tolerate.
Ethanol.
And we can talk about our favorite smells.
Yeah.
Great stuff.
Oh yeah.
Love that.
Yeah, bleach plus bleach plus 7X and
The warm room.
autoclaves.
That's right. Autoclave biological trash.
Yep.
Well, people who are not biologists will walk into a building where biology is done and they'll say, oh, yeah, this smells like a biology building. And I can walk into the same building. It's not a building I've ever been in before, and I don't smell it. I don't notice it.
There you go.
Autoclave E. coli.
Yeah.
Dickson, I want to point out, occasionally I take our little conversations before or after TWiV, and I stick them at the end of the episode.
Sure.
So I do that.
No, I didn't
now,
say...
you would not know this because you never listen, but that's fine. I'm okay with that.
They're called...
Yeah, right. He said holding a 45 Magnum in front of me.
They're called Easter eggs, Dickson, and in fact, that's what was on last week's show about me being sassy.
We had an 8-minute one, and some people really liked it.
Okay.
And I wanted to say...
I was just talking
we were
about the titles, that's all.
Back on plaque assays, purity of, noble agar versus agarose or whatever. Every time that our plaque assays went off and stopped working, we escalated the purity of the agarose that we get. So now we have, I think we only have about 2 more levels that we can go. So plaque assays just have to keep working.
Okay, so listen to this. We used to use noble agar, and I used to autoclave it and in batches, small batches, in
like
100 mil Gibco bottle,
and then age it in oak
and
barrels.
leave them on the shelf.
Yeah.
One time I came back after and then you just nuke one to liquefy it and then use it. One time I came back from a Christmas vacation. And I started doing plaque assays, and we did plaque assays where we always used 2 temperatures, or frequently used 2 temperatures, 31 and 40, and sometimes 37 to do mutants. And for weeks, the plaque assays, even with wild-type virus at 40 degrees, didn't work. I got no plaques. The cells were fine.
Eek.
Okay? I got no plaques. And this is one of these things, right, where it's not working. You go through everything.
Oh,
what's the matter?
Got a great story here, too.
Is it the serum? Is it the media? Is it the cells? You get out new virus, new cells. You make all this stuff.
Right.
It turned out, believe it or not, that the agar, if you left it on the shelf at room temperature for over 2 weeks, would poison a 40-degree plaque acid. It didn't kill the cells. It just poisoned the virus growth. Only if you left it on the shelf at room temperature for over 2 weeks.
That's weird.
It's because it's sterile and it's closed, right?
Yeah.
Pretty weird.
It's rearranging itself as we speak.
I don't know.
That's weird. Kathy, as far as escalating agar overlays, so we had one virus, rhinovirus type 2, which didn't work with anything. It didn't work with bacto agar, with any kind of agarose, C-plaque, low melt, high melt. Finally, we tried this highly purified agarose from Sigma. It worked. It costs $500 for 100 grams.
Yep. Yep.
Yow!
Type 6 or type 5 agarose. I don't know. I stopped working with that virus.
Because the cost
It's
is high.
ridiculous.
Too picky.
Fair.
Yeah.
Too
It's
picky.
just ridiculously expensive.
How funny.
Yeah.
It happens.
So the reason why I know that there's sulfur in the water in certain places was because I used to go up to Saranac Lake to work at the Trudeau Institute with somebody up there who was an immunologist. And I taught him how to handle the infection, Racaniello. And the way you do this is you infect rats, then you wait 30 days, then you skin and eviscerate the dead rat. You'd grind up the carcass, making ground meat out of it, and then
Ratomatic.
and then you'd put it into a large flask with 1% pepsin and 1% hydrochloric acid. And so, what do you do where is the water from? And from here, of course, it comes out of the tap, and it's a wonderful source of water that we have in New York City. If you do that in Saranac Lake, and we narrowed it down to the water eventually, it turns out that the water is loaded with sulfur. Nobody in Saranac Lake, you could eat a raw pig and you would never get Racaniello. As long as you drank water along with it.
Wow.
It killed all the larvae. It basically was a larvicide.
Why don't you use deionized?
Well, that's what we went to, of course. But
Okay.
here we didn't have to. We just used the tap water. It worked beautifully.
Yeah, there are all kinds of stories. We
Crazy.
could go. We could
Yeah,
collect.
that's right.
Send in your stories.
Send in your. Why not?